Hello, everyone. My name is Tamara Keene, and the study of my research this summer has been the effects of dietary fat intake on neuroinflammation in rats. To begin, I would like to direct your attention to each of these terms, dietary fat and neuroinflammation. Dietary fat is known to increase neuroinflammation. It is also known to increase impulsive behaviors. With that said, it has been the subject of our research to establish a correlation between impulsivity and neuroinflammation, but we don't know what that correlation looks like just yet. Impulsivity is assessed by behavioral tests, and neuroinflammation is assessed by immunohistochemistry. This is the subject of Dr. Cheatwood's expertise and our lab work this summer. Impulsivity and neuroinflammation are measured variables within this experiment, and diet is our changed variable. Half of our subjects will be given a high-fat diet, while the remaining half will be given a low-fat diet. What is the purpose of investigating neuroinflammation? Neuroinflammation will serve as a point of reference. It is our goal within this study to observe initial effects of diet on neuroinflammation, but also reverse possible cognitive impairments caused by the diet. We won't know if that reversal is successful unless we have easily comparable quantitative data. This will be given to us by investigating neuroinflammation specifically. Immunohistochemistry is a process in which we are using to investigate neuroinflammation, of which we are using two assays. Ionized calcium binding adapter molecule 1, or Iba1, is going to be our first assay, which will serve as a marker of microglia activation. Microglia activation is often ambiguous. We see that it is a proponent in the progression of diseases like multiple sclerosis and Alzheimer's disease, which work against our bodies. But microglia activation have been noted as beneficial in other areas, as in eliminated unused synaptic junctions within our brains that help increase cognitive functioning. Our second assay is a gliofibrillary acidic protein, or GFAP, assay, which indicates reactive astrocytosis. Reactive astrocytosis is a term not too many of us may be familiar with. This is just simply a condition following astrocyte injury that is noticeably different in shape and size. Oftentimes, the astrocytes will become larger. Our tissues of interest within this study are the hippocampus, which is displayed above in the tissue, that bolded region, the prefrontal cortex, the hypothalamus, and the dorsal striatum. Neurogenesis is another area of interest within our study. We are investigating neurogenesis by the number of Ki67 immunopositive cells within the hippocampus. It is important to note that Ki67 is a protein expressed on all new cells within the body. So this is a very non-specific test and will only give us baseline information for further investigation later. The Iba1 test results include as we see here, the green structures are microglia. There is more microglia in the high-fat brain tissue. Therefore, we can conclude that the higher microglia activation in high-fat brain tissue. Now, considering the GFAP results here, we aren't looking for the astrocytes, which are our green structures population as the previous test, but we are looking at the astrocyte morphology. And here we don't see any larger astrocytes. Therefore, we can conclude that there is no reactive astrocytosis within either tissue. At this time, we do not have any results for the Ki67 tests, as we're still in preliminary data collection phases. A special thank you to Lakshmi for being an amazing organizer throughout the summer and keeping us all on the top of our game. Dr. Cheatwood for allowing me to study within his laboratory this summer. Jason and Ellen for being amazing mentors and really increasing the personability of SRI. Bristy for being an amazing lab mentor and grad student throughout the summer. And Retta for keeping each of our programs in contact throughout the summer and hosting an amazing orientation week at the beginning of the summer. Thank you all.